My today's topic is Introduction to Medicinal Chemistry. First, in Unit 1, Medicinal Chemistry, subject code is BP402T in B Form 4th SEM. So, what is medicinal chemistry? In medicinal chemistry, the chemist attempt to design and synthesize a medicine or pharmaceutical agent which will benefit humanity. Such a compound could also called a drug. As a Latin arms, medicina, meaning the art of healing. It involved synthesis, SAR, that is structure activity relationship, receptor interaction, absorption, distribution, metabolism and excretion. The combined form is known as ADME. According to IUPAC name, medicinal chemistry, it concerns the discovery, the development, the identification and the interpretation of mode of action of biological active compound at the molecular level. Medicinal chemistry covers as the 3 stages, that is first stage is known as the discovery step. In discovery step involve the choice of therapeutic target, that is receptor, enzyme, target group, cellular, in vivo mode, in vivo model and identification or discovery of production of new active substances interacting with select targets. Such compound are known as lead compounds. And optimization step, which deals with improvement of lead compound. The optimization process uptakes primarily into account that increases in potency, selectivity and toxicity. The development stage, that is the 3rd stage, which purpose its continuation and the improvement of pharmacokinetic properties. History of medicinal chemistry. In 2000 BC, Materia Medica, that is 250 vegetable drug and 120 minerals drug are found. In 1500 BC, Egyptian Papyrus 700 drug is originated from animal, plants and minerals. Emperor Frederick II issued the Magna Carta in pharmacy in 1240. In synthesis of urea, that is 1828, started organic medicinal chemistry. Earliest as, that is side chain theory and chemotherapy and Fisher's look and key theory birth in modern medica, that is 1800. Medicinal chemistry received a formal recognition in academic pharmacy in 1932. The drug discovery. Drug discovery includes some following points, that is the discovery through small molecules, it forms through large molecules, through proteins and oligosaccharides, then it converted into new inorganic molecules and through the organic molecules. Objective of medicinal chemistry. Generally, we can identify the following stages of the drug discovery, design and the development. It may include 3 steps, that is the drug discovery, finding a lead, that is choose a disease, choose a drug, identify a bioassay, find a lead compound, isolate and purify the lead compound if necessary, determine the structure of the lead compound if necessary. Then drug design. In drug design we have to identify the structure SAR, that is structure activity relationship, identify the pharmacophore, improve target interaction, that is in pharmacodynamic, and improve pharmacokinetic properties. Drug development, that is its last stage. In drug development we have to concentrate on patent of the drug, carry out the preclinical trials, that is drug metabolism, toxicologic, formulation and stability test, pharmacology studies, etc. Design a manufacturing process, chemical and process development, carry out clinical trials, registered a market and the drug, make a money. Finally, medicinal chemistry includes synthetic and computational aspect of the study of existing drug and agents in the development of relation to the bioactivities, that is understanding SAR or isolating of the drug. What is combinatorial chemistry? A combinational chemistry is technique by which large numbers of different but structurally similar molecules are produced rapidly and submitted for pharmacological assay. These techniques use the same reaction condition with the same reaction vessel to produce the large number of analogues. That is, this technique is invented in the late of 1980s and the early of 1990s to enable the task to apply to many molecules simultaneously. Combinatorial library is the definition is collection of finally synthesized compound. Size, it depends on the number of the building block used per reaction and the number of reaction steps in which a new building block is introduced. Random libraries and focus on target libraries are random libraries are multiple libraries. Targeted libraries are templates of skillful libraries. Multiple targets in random libraries, one target in focused libraries. Highly deserved in random libraries. Focused library is highly structural and similarity. Mixture is found in random libraries. Single compounds may be found in focus or target libraries. Less than 5,000 compounds, greater than 5,000 compounds are found in focus or target libraries. Solid phase synthesis in solutions and solid phase. Non-purified compounds are found in random libraries and pure compounds are found in focus libraries. Non-purified compound and pure compound. On bad screening, if possible, screening is in solution. So application, these are some strategies of conventional and combinatorial, one molecule at a time and make purity test. In this, many molecule at a time make test and purity are found. 100 of molecules in a month, 1000 of molecule in a month. Slower lead generation, faster lead generation, high risk of failure, that is low risk of failure, synergy, and lead identification. Application where are very in application of combinational chemistry are wide. Scientists are used to create large population of molecules that can be screened efficiently by producing large, more diverse compound libraries. Companies increases the probability that they will find the noble compound of significant therapeutic and commercial value. Provide the stimulus for food, robot control and mobilization of strategies that allow high and multiple parallel approaches of the new drug discovery. Techniques in combinatorial chemistry. Solid support synthesis, that is on solid phase, such as resins bed, pins or chips. Next is split and mix method. Next is parallel synthesis, and other is solution phase synthesis. In this, solvent in the reaction flask is present. Solid phase synthesis is reactant are bound to polymeric surface and modified while still attached. Final product is released to the end point of synthesis. Example of solid phase supports is partially cross-linked polystyrene beds, hydrophobic in nature, causes problem in peptide synthesis due to the peptide folding. Shepherd's polyimide resins, that is more polar. Tentacles resins similar to environmental to ether to the THF, that is target essential resins, and beds, pins, functionalized glass surfaces. Method: beds must be able to swell in the solution in the solvent used and remain stable. Most reaction occur in the bed interior. These are the through this diagram we show that the resins in the bed are cross-linked, and through swelling it may be dispersed in different phases or different positions. Anchor or linker. A molecule might be which is conventionally attached to the solid support and which contains a reactive functional group. So allow the attachment of the first reactant, and the link must be stable and the reaction condition in synthesize but easily cleaved to the release of final compound. So differently linkers are available depending upon the functional group to the attached and the desired functional group on the product. Reasons are named and to define the linker, that is Merrifield and Wang ring. Solid phase synthesis or protecting group. A few protecting group used in solid phases, that is solid phase synthesis for amines, that is T-butoxycarbonyl, 9-fluorophil methyl methoxycarbonyl, 2-triethyl methyl methoxycarbonyl, or carboxylic acid, that is tetrabuesters, butyl esters, 9-fluoronyl methyl esters, 2-trimethyl ethyl esters. Parallel synthesis. To use standard synthetic route to produce range of analog with different analog in each reaction vessels, that is these reaction vessels are in form of vessels, tube or and well. They identify that each structure is known, useful for producing the range of analog, that is for SAR of drug optimization, produced for parallel synthesis, that is Houghton's T-bag procedures. Each T-bags contain bads and is labeled. Separate reactions are carried out on each T-bag. Combined T-bag are common reactions or workup procedures. A single product is synthesized within each T-bag. Different products are formed in different T-bags. Economic effort, example, that is combined T-bags by workshops, cheap and possible for any labs, manual procedures, and not suitable for producing large quantities of different products. Mixed combinatorial synthesis. To use the standard synthetic route to produce a large variety of different analogs where each reaction vessel of tube contain mixture of products. The identities of these structure in each vessels are not known with certainty, useful for finding a lead compound capable of synthesizing a large number of compounds quickly. Each mixer is tested for activity as a mixture of inactive mixtures stored in combinatorial libraries. Active mixtures are studied for further to identify active compound. This is some structure which shows the mix and split method. Example, synthesis of all possible dipeptides using 5 amino acids, and standard method would involve 25 separate synthesis, that is combinatorial procedures involve 5 separate synthesis, uses mixing of and split split strategy, that is glycine, alamine, phenylalamine, vanillin and serine, and 25 separate experiments should be formed. Quantitative structure relationship, that is QSAR, is a mathematical relationship in the form of equation between a biological activity and measurable physiochemical parameters. QSAR attempt to identify and quantify the physicochemical properties of the drug and to see whether of any properties have an effect of the drug on biological effect. The parameters used in QSAR is measured to potential the contribution group to the particular property of the parent drug. Activity is expressed as 1 log per C, that C is the minimum concentration required to cause defined biological response. Various parameters used in QSAR studies are lipophilic parameters, electronic parameters and steric parameters. In lipophilic parameters, partition coefficient, pi and substitution constant should be present. In electronic parameter, Hammett constant is present. In static parameters, Taft's constant and molar refractivity, where steric parameter is present. Lipophilic parameters, lipophilicity is partitioning of the compound between the aqueous and the non-aqueous phase. Partition coefficient, that is P is equal to drug in octanol upon drug in water, that is high P, that is high hydrophobicity. Pi substituent constant or hydrophobic constant are the pi substituent constant defined by Hansko and workers, measured by hydrophobic substance, that is relatively pi is equal to log P upon minus P upon log pH. Positive values implies substituents are more hydrophobic than H, and this benzene ring shows conversion through chlorine, that is chlorobenzene, and then it formed to benzamide. Through negative value implies imply substituents are less hydrophobic than H, and the Qsar equation may be included by both P and the small p. Capital P measures the importance of the molecule overall hydrophobicity that is relevant to absorption or binding, and P small p identify a specific region of the molecule which might interact with hydrophobic region. Steric substitution constant, it measures the bulkiness of the group and it represents its effect on the closeness of contact between the drug and the receptor site. It is much harder to quantitate. Taft's steric factor is measured by comparing the rates of hydrolysis of substitute aliphatic ester against standard ester under acidic condition. ES, which is represented by ES. Here ES is equal to log Kx minus log Co. Kx is represented by rate of hydrolysis of substituted ester, that is Kx, and Ko, that is co-represented by rate of hydrolysis of the parent ester. Molarity, molar refractivity, that is represented by Mr and is the measure of the volume occupied by an atom or group. Equation includes MW and density, that is the index of the refraction. Verloop steric parameter is computer program used for bond angle, that is van der Waals radii bond lengths. Big pharma drug discovery in the 21st century. The pharmaceutical industry in short of new drugs in second part of 20th century, about 50 to 60 new drugs were approved by FDA in every year in a constant. In 2002, a historical low of 18 drugs were approved by NCE. Later on in 2003, 2001, 2002 and 2003, 27 drugs are approved by FDA. In each and every year, various drugs were approved. Latest in 2014 and 2015, a new therapeutic biological product, that is anti-B is approved, and about 41 drugs have been approved by the new drug range, considering a high profile discovery which figure are unlikely to be dropped estimated by the recent drug system. In research newlets, the decline number of the new drug is based among other reasons on the current high therapeutic standard in many indications, focusing research on chronic disease such as cornea hurt, Alzheimer, arthritis, cancer and AIDS, as well as enhance the regulatory requirement for efficacy and the safety of the new drug. A lead can be characterized as a compound that has some desirable biological activity, not extremely polar or lipophilic, not contain toxic or reactive functional group, often molecular weight, as considered to be the most obvious characteristic of drug like lead. The lead should be also have a serious conjugates and that modulate biological activity, indicating that further structural modification will improve the selectivity and the potency. Medicinal chemistry Folcorel, earliest in medicine in 1500 years ago, 5100 years ago, Chinese Empress Shenyang, a book of hers, Pen Tensao Cheng San, contain alkaloids used today in the treatment of malaria and for fevers. Ma Hung contain epinephrine used as a heart stimulant for asthma, now used by bodybuilders to endurance athletes because it quickly converts fat into energy and increases strength to muscles fiber. Modern therapeutic is extract of foxglove plant cited by Welsh physician in 1250, used to treat dropsy, that is congestive heart failure, in 1785, contained digitoxin and dioxin, today called digitalis. Rational drug discovery. Nearly every modification of neurotransmitter, that is dopamine, serotonin, histamine, acetylcholine by classic Classical medicinal chemistry led to a compound with modified activity and selectivity. Steroid hormones modification led to similar success stories. In 1980s and 1990s, computer modeling of enzyme substrate complexes become a major driving force by a rational drug discovery and interpretation of SAR results. How should the medicinal chemistry respond? Historically, screening a black box, empirical SAR, but high relevance and guaranteed efficacy. Contemporary screening target isolation, precision, SAR, but relevance and efficacy unclear. The compromise takes secondary screening into the cellular context, that is still much skepticism, but primarily cellular screening really depend on the degree which the cells assay, reproduce and target diseases. So how do we blend efficacy lessons that passed underpinned by a network pharmacologic evidence with modern screening and secondary assay techniques? That is how much must we change our mindset. After all, we optimize activity of ADME, PK or more or less in parallel these days is an extra parallel target for two optimum quantum leap. So lead optimization where medicam gets through, that is balanced activities, balanced of physiochemical properties, balance of off-target activities. SAR activities relationship. In 1868, Brown and Fraser examined neuromuscular blocking effect of variety of simple quaternary ammonium salts to determine the quaternary amine in query was that cause for its muscles paralytic properties. Conclusion: the physiological action is function of chemical constitution. Structurally specific drug, that is most drug act as a specific site, that is receptor or enzyme. Activity, potency, susceptible to most small changes in structure. Structurally non-specific drug, that is no specific site of action, similar activities with varied structures, various gaseous anesthetics, sedatives and antiseptics. Example of SAR is sulfur drugs, that is an 1000 of analog synthesized lead sulfonamide, that is R is equal to H, and this sulfur drug is from clinical trial various analogs shown to possess three different activities, that is antimicrobial, diuretic and antidiabetic. SAR general structure of antimicrobial agents are group must be NH2, that is converted into NH2 in vivo positions. Replacements of benzene ring or added substituent decreases or abolish the activity. Halogen group must be added or maybe removed out, so R can be usually added or reduced, but its potency may be reduced. Thank you everyone. Here are some views and my points on medicinal chemistry. Thank you.